Hello, welcome to your own channel, Curious Corners in Biology, where we inquire into this fascinating subject of biology with an element of scientific inquiry. And today we will be continuing with the topic of hematology. And today we are going to discuss something very interesting, that is about blood groups. So you might be thinking we already know what are blood groups. No, we are not going to discuss the system of blood group which is already known. We are going to discuss rare blood groups. Okay. So it has been known that more than 30 different blood groups are seen in humans. Okay. It's so fascinating. And it simply means that there are more than 30 different options for even insects to bleed humans dry. Okay. Jokes apart, let us first think about the most common blood group system, which is called as the ABO blood group system. ABO blood group system was discovered in 1900s by Karl Landsteiner, and there is a very basic concept about this blood group system, which you might be familiar, is that A blood group will have A antigen and B antibody. The antibody cannot be same as the antigen, otherwise there would be an agglutination reaction. Then B blood group will have B antigen and A antibody, and then there is O blood group having no antigen and so both the antibodies, that is antibody A and antibody B. There is also AB blood group system which has both antigens A and B but no antibody. So, as per this blood group system, O blood group is considered as universal donor and AB blood group is considered as universal recipient. Now, you might have known that these blood groups are also positive and negative. It is simply based on presence and absence of Rh factor. There is a specific antigen from rhesus monkey which was discovered to be present on RBC membrane, and if that's present, that is Rhd antigen, if that's present, then any blood group A, B, AB or O becomes positive, and if that's absent, then ABO blood group will become negative. Okay, that is A negative, B negative, AB negative and O negative. So far so good. Now let us think about certain rare blood groups. So I'll be sharing my slides and hope this will be visible to you. Okay, so the first rare blood group which we are going to talk about is Duffy blood group. Okay, so this is named after a subject, that is a patient, Richard Duffy. So in this blood group, in the blood of Richard Duffy, Duffy antigen was discovered. This is also called as F5 glycoprotein, and this is encoded by ACKR1 gene. Now this is a blood group in which the antigen which is present on the RBC membrane is also acting as a receptor for malarial parasite, that is different species and strains of Plasmodium, like Plasmodium vivax, Plasmodium knowlesi, Plasmodium cynomolgi. Okay, so this is a unique thing about this blood group, right? And the clinical significance is that this Duffy antigen F5 protein is associated with many health risks like asthma, transplant rejection. Of course, antigens on RBC membrane play a very important role in transplant acceptance or rejection. Ultimately, any transplant eventually gets rejected. For example, lung transplant or kidney transplant. It is also associated with low neutrophil count, neutrophil being a type of WBC. Endotoxin response. In this blood group, it has been seen that the levels of endotoxin in blood cells would increase, HIV infection as well, inflammation, Multiple myeloma. Multiple myeloma is cancer of plasma cells. Plasma cells are a kind of WBCs in our blood. Hemolytic disease of the newborn. So in our discussion, this word will be very common. Autoimmune anemia, hemolytic disease of newborn, transfusion reaction. What do we mean in general by these terms is that ultimately body's RBCs, which are own RBCs, are attacked by certain antibodies. If that's the case with a newborn, then maternal antibodies would attack the RBCs of fetus, which is called hemolytic disease of the newborn. This leads to severe anemia, jaundice, and eventual death of the fetus within the womb of the mother. And so hemolytic disease of newborn is also called as erythroblastosis fetalis. Transfusion reaction is any sort of blood transfusion response which may be triggered by production of antibodies against the antigen of the donor blood. Antibodies will be produced by recipient's blood. Sickle cell anemia, RBCs will become sickle shaped. Okay. Then we have KEL blood group. Now this is also a unique antigen for a blood group system called KEL group. And these antigens are present on RBC membrane, and these are also target for autoimmunity. That is target for WBCs which can destroy RBCs as autoimmune reaction. So of course, KEL antigens are responsible for transfusion rejection, autoimmune hemolytic anemia. Autoimmune hemolytic anemia differs from hemolytic disease of the newborn. In autoimmune condition, even in an adult body, the immune system would attack self RBCs or body's own RBC. And also hemolytic disease of newborn is associated with this blood group. When does these clinical complications develop? During blood transfusion. Okay. Then we have Lutheran blood group. So this is classified based on presence of Lutheran antigens, and there are 19 of those. If those are seen, then this becomes a unique blood group, Lutheran blood group. But this is seldom the cause for erythroblastosis fetalis or any kind of transfusion reaction. Mainly this has been found in African population, Lutheran blood group. Then we have KID blood group. Now, interesting thing about this blood group is that there is a rare blood group associated with this KID blood group, and that rare blood group is called as KID blood group, and that is called as KID null phenotype. That is called KID null phenotype. Okay. So, in this, we have a specialized antigen under consideration that is called JK antigen. Now, this JK antigen is present on the surface of RBC membrane, and this antigen is present on a protein which acts as urea transporter for RBCs. Now, please understand that when this antigen is associated with urea transporter, that means this antigen is associated with transport of urea inside and outside RBCs. So when blood reaches the kidney, concentration of urea is dependent on this antigen because it is associated with urea transporter protein. So this antigen is associated for docking urea in the kidney, so thereby increasing the concentration of urea in the kidney, and hence urea is flushed out in the urine. Now, if this antigen is absent, that is in KID null blood group, then there is less ability of the kidney to concentrate urea in the urine. But this is not associated with any severe manifestation. For KID null phenotype, so to say, But then kid null phenotype is an extremely rare phenotype. Okay. So of course, this blood group is important in transfusion medicine. All right. Then we have Bombay phenotype. So this has currently and recently come under discussion that this is a unique blood group which was found in Mumbai in India, and this is only found at an incidence of 1 in 10,000 individuals in India and 1 in a million people in Europe. Okay. Now, think about ABO blood group system, and in that, a specialized antigen on the surface of RBC, that is H antigen, is responsible for production of A antigen, B antigen. Now, if H antigen is absent, then A antigen and B antigen is not produced, and this unique blood group is called Bombay phenotype. Okay, and please remember that this is not O blood group, because in O blood group H antigen is present but it is not expressed into A and B antigen. But in Bombay phenotype H antigen is altogether absent. So if it is altogether absent, it is not O blood group. It is not O blood group. Okay. So there is a difference between O blood group and Bombay phenotype. All right. Then we have Diago blood group. So Diago blood group is again present on RBC membrane. It is associated with a specialized protein on RBC membrane that is called band 3 protein. And this antigen along with the protein has an important function of chloride or bicarbonate exchanger. We know that chloride shift and bicarbonate in RBCs are associated with carbon dioxide transport, and this phase occurs when blood is moving from different tissues towards the lungs. Okay, so from lungs, CO2 will dissociate from RBC and then it is let out from the body. Okay, and this antigen is also associated with kidney, and it is associated with a specialized condition which can develop when this is deficient, that is called renal tubular acidosis. First of all, this blood group is rare, and if the antigen associated with this blood group is mutated or if it is defective, then it leads to abnormality along the RBC membrane. That means the shape of the RBC will be distorted. Okay. And then at the level of kidneys will be defective in secreting acids into urine. So if they are unable to secrete acids in urine, acid buildup would result in blood, which is called renal tubular acidosis, which is connected with the mutation with the antigen associated with this blood group. So one of the mutation causes hereditary spherocytosis, which is distortion of RBC membrane, that is shape of RBC. There is another variant to this disorder that is called as hereditary ovalocytosis or elliptocytosis. This is associated with abnormal RBC membrane, and this is sufficient to cause hemolytic anemia, jaundice, and splenomegaly. Splenomegaly is enlargement of spleen. Okay, due to abnormal RBC shapes, because some sort of autoimmune reaction or response is also induced due to abnormal RBC shape. Okay, so this is Diago blood group. Then we have MNS blood group. MNS blood group is also rare, although it is present in Caucasians, it is present in Africans, Asians. But this blood group was discovered in 1927 after the discovery of AB blood group system. And it is associated with sugar-bearing proteins called as glycophorins found on the surface of RBC membrane. Okay. So glycophorin is divided into two portions, one of which is associated with the underlying cell, that is, it is connected with the RBC membrane, and the other part or the portion of glycophorin is connected with sugar. And this is how MNS antigens are formed. Now, M and N antigen were discovered first, and S antigens were discovered 20 years later after the discovery of M and N antigen. Okay. Then we have Lewis blood group. So this was named after the discovery in a subject named Lewis. So she was a female, and it was found that her body produced antibodies against antigens found on her RBC membrane. And also antibodies were produced against antigens which were found surprisingly in body fluids like saliva, gastric juice. So that means antigens associated with this blood group are also present in secretions of the body. Okay. So Lewis antigens were named after this lady, and such antigens are also called as secretor antigens or secretory antigens due to their association with secretory body fluids. And of course, Lewis antigens are associated with retarded growth, and it is also cause for transfusion reactions sometimes. Then there is P blood group. Okay, this is also a rare blood group system, and doctors of Manipal identified this penal phenotype for the first time in India, and it is made up of three kinds of antigens, P, P1 and PK antigens on the surface of RBCs. These antigens are also associated with the epithelial cells of the urinary system, and these antigens serve as sites for attachment of E. coli, Escherichia coli bacteria. And Escherichia coli is a cause for UTI, that is urinary tract infections. So these antigens are associated with UTI as they serve as sites for binding or adhesion of E. coli. Okay. These are also associated with acute intravascular hemolytic reaction, also associated with severe erythroblastoma fetalis, and of course, this severe erythroblastoma fetalis can cause spontaneous abortion in case of pregnant females. Okay. Then we have XG blood group system. So here, of course, XG antigens are specialized antigens associated on the surface of RBCs. Okay. And surprisingly, these antigens are connected with X chromosome. That means they are encoded by the genes of X chromosome. Okay. So it has been found that if son has XG blood group, then son receives these antigens only from mother, but a daughter receives these antigens from mother mainly because she has X chromosomes, but son only receives from mother. Okay. There is a whole genetics associated with XG blood group system. Okay. And XG blood group is having more incidence in daughters of XG parents than sons because son also has Y chromosome. So this antigen is developed at birth, but it is not much associated with transfusion or hemolytic responses. It is not inducing instant response of antibodies. Okay. Then we have golden blood group. This is very interesting. Now, this word golden blood group does not mean that the color of the blood is golden. No. Actually, this golden word is connected with the rarity of this blood group. Given the fact how rare is this blood group in terms of its occurrence. Okay. So, at a rate of 1 per 1,000 people or even fewer than that. So, this is rarest of the rare blood type, and fewer than 50 people in the world will have this phenotype. So that is why those are golden people having golden blood group. So golden is connected with rarity. Okay. Now, please understand that when we talk about RH factor, we are talking about an array of RH antigens, and there are 61 RH antigens. Out of those, if RHD is present in the blood, then blood group will become positive, and if it is absent, then blood group will become negative as per ABO blood group system. But here, no RH antigen of the 61 RH antigens is present. So RH antigen is nil. None of the 61 RH antigens are present in golden blood group. Now, this is different from RH negative blood group because RH negative will not have RHD antigen, but the other 60 antigens may be present in RH negative blood group. But in golden blood group, no RH antigen is present. Okay. So, of course, this is important in the medical world, and this can be used as a universal donor if there are some rare blood groups, and if a person having rare blood group is in need for blood transfusion, then this golden blood group can be utilized. But then only 50 people, 50% people of the world are having this blood group. So that's also a fact. So yeah, that much for today. Hope you have enjoyed this. And if you are interested in such content, then please subscribe to this channel. This is very interesting and unique of a kind channel in which we discuss every possible unanswered question in biology. And the mission is very simple, to ignite curiosity in the subject of biology. This is what this channel is meant for. And please check out this book, Curious Corners in Biology. You can click on the link given in the description, that is available in Amazon Kindle format. Please check out, and it's a unique book. It is not a textbook. It is a jargon of questions, questions which are rare from every nook and corner in the subject of biology, from every unit of biology or any topic of biology. Okay. So this is not just known literature which we are discussing. We are trying to discuss questions which demand reasoning, which demand scientific inquiry, which demand critical or abstract thinking. Okay. So that is all for today, and see you soon. Till then, bye.